Today we will continue the staining technique. Today we are going to study the gram staining technique. I am Dr. Sharad Deshmukh from SS Jaiswal College, Arjuni Morgaon. Now let us see about the gram staining technique. This staining technique is most widely used as a differential staining in the microbiology because it differentiates the bacteria into two main groups, that is gram positive and gram negative bacteria. Hence it is referred as the differential staining. In this case, we'll see the details about the gram staining. Now, who discovered this technique? This technique was first introduced by Hans Christian Gram. He was the Danish physician who devised the gram staining technique in 1883 while working in the Berlin. Later, he published this procedure in 1884. At that time, he was studying the lung tissue sections from the patients who had died due to the pneumonia, and he found the bacteria which are stained by the gram staining technique. In this staining technique, he formulated the criteria as the primary stain by aniline junction violet, then ligolcydine as a mordant, and he used the absolute alcohol as a decolorizer, and in the final step he used the Bismarck brown. While the Carl Weigert has done some modification in the gram staining technique, he was from the Frankfurt and added the final step of the staining with the safranin instead of the Bismarck brown. In the original procedure of the Christian Gram, in his paper, Dr. Gram described how he was able to visualize what we know now or call the Streptococcus, Bacillus, Clostridia, and various bacteria which are observed by him in histological sections. But he did not actually use the safranin as a counter stain in the original procedures. That is, to stain the gram negative cell would be the colorless in this technique. He instead recommended using the Bismarck brown as a counter stain to enable the tissue cells nuclei to be visualized instead of visualizing the gram negative cell. Now we'll see the principle behind the gram staining technique. Firstly, you can see here when the bacterial smear is stained with the crystal violet and iodine, some bacteria will get stained, all bacteria will get stained with the crystal violet and iodine, but there are some bacteria which will resist the decolorization by alcohol in the decolorization step, and they will appear violet in color, and these are referred as the gram positive bacteria. In contrast, there are some bacteria, they get decolorized with the alcohol, they will lose the crystal violet and iodine complex, and they get stained with the counter stain safranin and appear red in color, are called as the gram negative bacteria. This is the basic principle behind the gram staining. Now let us see the mechanism why the gram positive appear violet in color and the gram negative appear as a red in color. This is the basic mechanism behind the gram staining. There are various concepts for explaining the why gram positive bacteria retain the crystal violet and why the gram negative bacteria they retain the counter stain. One of the concept is the gram reaction is dependent on the permeability of the bacterial cell wall and the cytoplasmic membrane to the diiodine complex. So according to this principle, that they are depending on the permeability of the cytoplasmic membrane and the cell wall. In the gram positive bacteria, the crystal violet diiodine complex combines and to form a larger molecules which precipitate within the cells. While the process of decolorization by alcohol, they cause the dehydration of the multi-layered molecules and causing the cell wall to trap the crystal violet iodine complex within the cell. Hence, they retain the primary stain, and the gram-positive bacteria don't get decolorized, and they will retain primary diiodine and appearing the violet in color. There is another concept that the gram-positive bacteria have the more acidic cytoplasm, and hence they have the more affection toward the basic dyes and bind to the basic dye like crystal violet with more firmly, and they retain the crystal violet and iodine complex. This is also one of the concepts that explain why the gram positive appear violet in color. Let us see this procedure, why the gram positive retain the crystal violet. You can see here the bacterial cell, they are possessing the cell wall, and we are staining primarily with the crystal violet. When you add the crystal violet, they will get stained the cell violet in color, and in the next step we are adding the iodine, and it will form the diiodine complex, and the bacteria will get stained. All the bacterial cell, whatever it may be, the gram positive or gram negative, they will stain with the crystal violet and iodine complex. We'll see about the particularly the gram positive bacteria when we add the alcohol as a decolorizer. Now when we add the alcohol as a decolorizer, they do not get decolorized by the alcohol. Instead, they retain the crystal violet diiodine complex, and due to the shrinkage of the cell wall, and they will retain the violet color and appear the violet in color. That is the gram positive bacteria. In contrast, in the gram negative bacteria, the alcohol being a lipid solvent, and that will dissolve the outer lipopolysaccharide membrane of the cell wall, and they will also damage the cytoplasmic membrane to which the peptide glycan is attached. That is, the concept depend on the lipid content. That is, the lipid content is more in gram negative as compared to the gram positive bacteria, and as a result the diiodine complex is not retained within the cell, and that permeates out the process of the decolorization, that will lose the crystal violet and iodine complex. Hence, when the counter stain is added, now they will take the counter stain, that is the color of the counter stain, and appear pink in color. So we have seen that the gram positive will retain the crystal violet iodine complex, they appear violet in color, and the gram negative will get decolorized and they take the counter stain and appear pink in color. This is the basic mechanism behind the gram staining technique. So you can see here in the case of the gram negative bacteria, we have already stained with the crystal violet and iodine complex, and when we add the alcohol as a decolorizer, and this will get decolorized easily, they will lose the crystal violet and iodine complex, and they get decolorized totally. And now they will stain with the counter stain safranin when you add in the last step, and the cell gets stained red or the pink in color, and they will appear the red in color. So gram positive as a violet color and gram negative will appear red in color. This is the basic mechanism behind the gram staining. In the next lecture, we'll see the complete steps which are involved in the gram staining technique. Thank you.
Subscribe to the channel for more videos related to microbiology.